It's no secret that we had two titans of adult sickle cell disease in this state for several decades. Ever since Bob Adams recruited Dr. Kutlar down almost 2.5 decades ago, he has run a phenomenal sickle cell program at MCG. And even after all these years, it's hard for me to think of it as anything other than MCG. He's been a remarkably productive and perennially well-funded investigator in sickle cell disease with many signatures, but perhaps his greatest signature is his work on hemoglobin variants and fetal hemoglobin. And he has really been a wonderful colleague, friend, and collaborator for Jim. As we've had two of the epicenters of really treatment of sickle cell disease in the state of Georgia. It's interesting, we had two centers of excellence in sickle cell disease in Georgia, and arguably no center of excellence in cancer until only a few years ago. In fact, I think for a while, between Abdullah and Jim, their funding dwarfed all cancer funding in the state. So it's a pleasure to invite our good friend and partner and colleague of many years, Dr. Abdullah Kutlar, who's going to talk about lessons learned in the past 4 decades when he started working on sickle cell at age 9.
Thank you, Fadla. I am not that premature, though. I would very much like to thank the organizers for having me here. It's a great honor for me to speak at a symposium honoring my good friend and colleague Jim Ekman. We go back a long way. I won't say how long, but I first met Jim when I was a novice in Titus Heisman's group at then MCG. And many of you, I'm sure, know Titus Heisman or remember Titus Heisman. Jim certainly does very well. He was a terrific protein chemist, and at the time of his retirement, his laboratory had characterized almost 1/3 of the then known hemoglobin variants. And he also had an interest in sickle cell disease and held 3 cycles of the comprehensive center grants. He was also one of the old school autocratic European professors, and that autocracy permeated all areas of life, in that not only in your work, but you couldn't like anybody that he was mad at, and then you would be in serious trouble. And that happened with Jim a few times. But after he was out of the scene, and I think our relationship with Jim and the Grady Emory group has been nothing but collegial and very warm. It's been a terrific symposium, and I am in the presence of great investigators so far. I've enjoyed the great talks. I'm going to take a little different approach and kind of approach this from the perspective of what we have learned in the past 4 decades with clinical trials. Let me start by citing some of the milestones that is very well known to this audience. I'm not going to go back to the days of Jim Herrick, which we had the centennial short time ago, but I think everybody would agree that we learned a lot from the cooperative study of sickle cell disease in terms of the natural history of disease. This was followed by another landmark study that was a big impetus on newborn screening, in which Jim has been phenomenally active and impressive. That was the prophylactic penicillin study, which showed that penicillin prophylaxis after newborn diagnosis of sickle cell disease did save lives. The other study that was important, a landmark MSH study, of which both Emory, Grady, and we were part of, and it ended up in the approval of hydroxyurea as the only known disease-modifying agent today. And I think it was shown in Alan's slide that 2 follow-up studies published in JAMA and American Journal of Hematology also hinted that use of hydroxyurea was associated with an improved survival in this patient population. Then comes STOP and STOP 2 by my colleague Bob Adams, They were both stopped early, but for different reasons. Stop 1 for a positive result in that screening with TCD and initiation of prophylactic transfusions in patients who were deemed to be at high risk because of high TCD velocities did prevent strokes, but the stop 2 was also stopped short because it showed that at the current state of knowledge of the pathophysiology of cerebrovascular disease and sickle cell, we could not discontinue transfusions in this patient population. And then, similar to the MSH study, Baby Hug study was published in 2011, we heard from Danny this morning that one of the most promising results of the Baby Hug was the protective effect of hydroxyurea on neurocognitive function in these very young babies and infants. And as a result of this, I think Alan showed a similar graph. The life expectancy in sickle cell rose from around teens in 1970s when the National Sickle Cell Prevention Act was passed to about 50s in 2000 and 2010. Now, this is well known to everybody, but one of the problems that I have struggled with ever since I've been in this field is the number of sickle cell disease patients. And for many decades we said there were 70,000, but the closest estimate was given by our colleague Kathy Hassel with meticulous work through these different centers, and she estimates this number to be somewhere in 110,000. We know that there are approximately 3,000 newborns. Life expectancy can be said to be in the mid-50s now, and overall survival at 18 years of age, this is the good news part of Charles Quinn's study, is about 94%, but then acute chest syndrome and multi-organ failure continue to be the #1 cause of death, and sadly for us adult providers, most of the deaths occur shortly after transitioning these patients from pediatric to adult care, which I think says a lot in terms of the adult care. This is a different rendition of what Clint showed. I won't elaborate too much on that. And again, I want to point out that hemoglobin S polymerization is the center of the phenomena, but I think many people in this audience, particularly Bob Hebel, Clint himself, and others contributed a lot to the downstream effects, better understanding of downstream effects, and perhaps identifying these downstream effects as potential therapeutic targets for sickle cell disease. And for the sake of time, I won't go over each one of these, but suffice to say inflammation, coagulation, red cell dehydration, as very eloquently described by Clint, all contribute to disease pathophysiology. And a summary slide, I think what we know is that clinical spectrum of clinical complications differs from pediatric to the adult age, in that in pediatrics we deal mostly with acute episodic problems, and as the patients age to adulthood, the picture becomes dominated by various chronic organ failure or chronic organ damage syndromes. And then, of course, vaso-occlusive episodes, acute chest, and some other episodic acute events continue to be present in both the adult and pediatric setting. In terms of addressing these, unfortunately, we can't boast a large therapeutic armamentarium these days. We have transfusions, both episodic or chronic, and we know that the best indication for chronic transfusions is in the primary and secondary stroke prevention. Episodic transfusions can be life-saving, but we also know from the studies of iron overload that it can be a cause of great iatrogenic pathology in this disease. Hydroxyurea is the only approved agent for hemoglobin F induction, and then our success in symptomatic management of the disease, especially pain management and IV fluids, I would say overall leaves much to desire, despite the pioneering work that Jim did in his center in terms of optimizing this management. And we will hear from Mark Walters on the only curative approach to this disease, which is stem cell transplant. Now, having viewed what we have currently, then let's take a quick look at the potential therapeutic targets in light of what we know about the disease pathophysiology. We talked about supportive and symptomatic measures. The anti-sickling therapies are, of course, an important centerpiece of all therapeutic strategies. Hemoglobin F induction, by virtue of gamma chain's potent anti-sickling effect, is a very important goal. I'll come back to that a little bit. But then modulation of oxygen affinity to prevent deoxyhemoglobin S polymerization is emerging as another potential therapeutic means, and there is this high oxygen affinity variant that Greg Cato has been developing. I'm hoping that we will hear more about that in the upcoming ASH meeting. And then the adenosine signaling pathway, primarily through 2,3-BPG, by altering the oxygen affinity of hemoglobin and trying to keep the hemoglobin S in the oxy state rather than becoming deoxy, is an effective, hopefully effective anti-sickling strategy. Clint described elaborately the cation loss and dehydration phenomenon, so I won't go long, and they clearly have a place, and followed by this, there's anti-adhesion therapies. There are various agents that have been used for P-selectin blockade or pan-selectin inhibition, IVIG and beta-adrenergic blockade, and then finally anti-inflammatory therapies, corticosteroids, which have been shown to be effective, but the downside of this is that with cessation of therapy, there is a high rate of rebound, both in crisis and acute chest syndrome. IMiDs are potentially interesting agents. I will come back to that. And anticoagulation therapy with low molecular weight heparins. There's a paper published from Saudi Arabia which showed that by the use of low molecular weight heparins, the duration of hospitalization from a painful episode and opioid usage can be decreased. But that, again, remains to be seen. Mentioned hemoglobin F induction, hydroxyurea is the only approved agent. Then there are a group of other agents that are in early trials, which include histone deacetylase inhibitors for one, epigenetic modification, bitrate derivatives, and others. We have a trial with panobinostat or LBH589 that's ongoing, and DNA methyltransferase 1 inhibitors have been shown to be effective, especially in hydroxyurea refractory patients, and thalidomide derivatives. Thalidomide has been used not so much in sickle cell, but there are case reports of its successful use and successful induction of fetal hemoglobin in refractory thal major patients, 2 from Italy and 1 from Mexico. And pomalidomide is something that we have just completed a trial, which I will come back to. Now, our knowledge in terms of regulation of fetal hemoglobin has really increased in the past 5 or 6 years, and although 10 years of genetic association studies unfortunately have proven to be disappointing, and I have personally lost a lot of interest in that. Nevertheless, suffice it to say that at least in the area of fetal hemoglobin regulation, we've had some positive and encouraging results. Now, there are 3 loci that have been shown to be important in regulating fetal hemoglobin. One is BCL11A. The second one is an intergenic region on chromosome 6q between HBS1L and MIP. And then, of course, the third is the beta globin locus, especially the so-called XMN1 site in the G gamma promoter. And it is widely believed that variation in these 3 sites account for about 50% of variations in hemoglobin F levels. And finally, erythroid-specific transcription factor, KLF1, which used to be known as EKLF, which binds the beta promoter, is also a strong regulator of BCL11A, and BCL11A is regulated by this KLF1, and there is a paper that was published in Nature Genetics, which showed that haploinsufficiency of KLF1 resulting from a nonsense mutation in the gene resulted in a phenotype of hereditary persistence of fetal hemoglobin. And then again, of course, Stu Orkin's group was able to generate a erythroid-specific knockout of BCL11A, because if you knock out BCL11A in stem cells, it's lethal due to its important role in brain and B cell development, but erythroid-specific knockout of BCL11A results in almost a cure of transgenic sickle mice with very high levels of fetal hemoglobin, which is evenly distributed in red cells. So all of this put together, the regulatory scheme for fetal hemoglobin is that KLF1 is the regulator. It acts in two ways: by increasing BCL11A, which with this suppressor complex binds between the gamma beta intergenic region and suppresses gamma expression, and then on the other hand it also binds to the beta promoter, increasing the expression of the adult globin gene and thus playing a major role in the switch. Well, having hydroxyurea for these many years, why do we need other hemoglobin F inducers? There's several reasons. You know that hydroxyurea is not curative in the highest responders. They still have complications, and we published in 2005 that patients on hydroxyurea still die of sickle cell complications. There is a wide belief in the literature that about 30% are non-responders. I am not a believer in that. I think, as Russell Ware put it, there are more non-swallowers than non-responders, and I think this is mostly a matter of non-compliance. And again, but it's also true that hydroxyurea is not protective against every complication of sickle cell disease, avascular necrosis and leg ulcers being just two of them. So with this, I'm going to show some encouraging data that we had on pomalidomide. 6 years ago, I had an interest in using IMiDs purely as anti-inflammatory agents in sickle cell disease. And then I proposed to use lenalidomide, but that was at the time when lenalidomide was just approved for MDS, and the company said, no, we're not interested, go away. So 2 years later, they contacted me and said, are you still interested in pursuing IMiDs in sickle cells? I said, yes. And then they showed me this data that was published by one of their scientists in JCI. She tested bitrate, hydroxyurea, and pomalidomide and lenalidomide in primary erythroid cell cultures to see their effects, and showed that pomalidomide was the most potent inducer in the system compared to even hydroxyurea and others. This is the green line. Furthermore, the response was dose dependent, but also if you went up to high molar concentrations of hydroxyurea and butyrate, there was a drop primarily resulting from cytotoxicity, cell kill, which was not the case with pomalidomide. So then we got together in two centers, including ours, and Dr. Myler is in the audience, who pioneered the mouse work on this, which I will show here. He showed that in Towns transgenic mouse models, pomalidomide induced fetal hemoglobin in a degree that's comparable to hydroxyurea. And this was accompanied by an increase in total hemoglobin also. But the combination, either in low or high doses, did not have that same effect. It was flat. And again, this is Stefan's data published in Blood in 2011. It shows nicely that pomalidomide increases marrow cellularity, as shown here, as opposed to hydroxyurea, which is myelosuppressive, of course. And most of this increase results from enhancement of erythropoiesis, as clearly indicated here by a decrease in ME ratios. So with that, we launched a clinical trial in two centers between Wayne State and ours, and this was a phase 1 trial looking at dose-dependent tolerability of pomalidomide. It was 0.5, 1, 2, 3, and 4, and here I'm showing the doses of 4 milligram cohorts. 2 out of 3 patients responded, and 1 patient went back on a 3 milligram dose because of the perceived clinical benefit, which was in his case healing of his leg ulcers. and at about a year and a half, his fetal hemoglobin doubled, and his F cells went up to as high as 85.7%. So it was quite a gratifying response, and with that now we're trying to launch a phase 2 trial of this agent. Now, if you look at clinicaltrials.gov, there is a large number of trials in sickle cell, and 139 are still enrolling. This is very encouraging based upon what we had 2 or 3 decades ago. But what I want to do is focus on some of the clinical trials which Clint alluded to in his talk, especially with negative results, and try to discuss why these trials are negative. And this is strictly my editorial comments and is open to very vigorous discussion, disagreement, and argument. So I will look forward to that. Well, one of them was flow course study, which was published in JAMA in 2001. It was a promising agent that improved viscosity, but it was negative. The second study, Clint went into detail. Senecapop phase 3 trial was cut short despite some hematologic benefits because of the downside, which was an increase in pain. And then there was nitric oxide-based trials, biomarin and inhaled nitric oxide. These were all negative, especially the Gladwin study, which was a very well-designed and conducted study, did not show any effect. And then finally, we tried Steve Embury's compound in sickle cell, and it was also cut short, although we did see some effects in terms of biomarkers. So why do these trials fail? Well, as Clint went into detail for Senecapop, and this is the case in many others, I think these are mostly based on sound and validated preclinical studies and data. And among these, we know the importance of adhesion in sickle cell pathogenesis or complications, role of cation loss, and nitric oxide depletion. The study design for most of them was sound, and many trials reached their accrual goals. So what was the reason? Could it be endpoints? What was the endpoint in this? I think pain was the endpoint that was associated with most failures. And of course, the other one was targeting phenomena solely downstream from the sickling and targeting a Sickle mechanism in vaso-occlusive crisis and composite endpoints. I'll try to illustrate these one by one. Why is pain a risky endpoint in sickle cell disease? Pain is a very complex phenomenon, and we're just beginning to understand the different mechanisms of pain. It's not just vaso-occlusion. I believe that there's a transition from acute episodic nociceptive pain by age to more chronic neuropathic and centralized pain, and when you get to that stage, it becomes very difficult to take pain and evaluate that as a sole endpoint. Well, you know, in Wally Smith's Pisces study, which I think is a very important study, we know now that 50% of sickle cell patients experience chronic pain. And in some of our studies, I'll show data that self-reported pain does not always correlate with other measures of pain. For example, pain pressure threshold. And this is the summary of the Pisces study. Don't have time to go into details. Pablo, I have 5 more minutes. But again, it was important to change our concept, again, among many other studies that had been previously published, that sickle cell disease people are fine, they just have crisis. When they recover from that crisis, they're okay. No, they're not okay. We know that they're not okay in terms of the ongoing inflammation, anemia, hemolysis, but they're not also okay, especially adults, from the perspective of pain. A lot of them do continue to experience chronic pain. Now, when I show this slide, I have to make sure that I'm not in violation of HIPAA, because these are not actual patients. These are our research coordinators. Some of you may know them. They're showing here a pressure pain algometer, which we used in one of our studies in the P20 application. And typically, there are 3 areas that are tested, as shown here: masseter, trapezius, and ulna. What happens in this case is the operator puts pressure, the algometer measures it, and it's hooked up to a laptop. Laptop shows the rate of pressure increase, and the patient has a button in his or her hand and is asked to press the button when the pressure sensation changes to pain. And that is recorded as kilopascals or different parameters. So this is the tracing of the computer. When we looked at this pain in different states, this is masseter. It's obvious that sickle cell patients have a much lower pain threshold in terms of kilopascals than the non-sickle cell controls, and this is statistically significant. This holds true for trapezius and especially for ulna. So at all three sites, sickle cell patients are hyperalgesic. Now, let me emphasize that these measurements were done at the so-called steady state, when they were not in pain or when they were 4 weeks away from a known pain crisis. And those who developed pain crisis in the ensuing 2 weeks were censored, so they're not included in this analysis. Now, moreover, I think we see a very good correlation of hyperalgesia between different sites, especially more so in sickle cell disease with very high R and P values than control. So it's not that these measurements are not correlated or specific to one particular site. So they're globally hyperalgesic, if you will. Now, this speaks to the risk of taking pain as a sole endpoint in sickle cell disease, especially in adult patients. When I mentioned this, some of the venerable pain researchers in a previous meeting were upset because this was construed as my advocating not to take pain as an important endpoint. To the contrary, I do think that pain is very important, but I am trying to point out here that pain is not as simple in sickle cell disease as we thought it is. A case in point is a recent publication that appeared in September in Blood by Kalpana Gupta and her colleagues in Minnesota that showed a contribution by neurogenic inflammation triggered by mast cells, which I think is a very important and seminal observation. So a lot is going on. So the other point is that targeting phenomena that are downstream from sickling can be risky. Flocor targeted viscosity and adhesion. Icogen studied the Senecopa targeted RBC dehydration, and the two studies targeted nitric oxide bioavailability, and our study was geared towards P-selectin inhibition. Again, similarly, if you take one specific target in a pain crisis, because of the very complex pathophysiology of a pain crisis, of which we unfortunately do not have a full understanding, that sets the stage for failure in many studies. Now, an example of failure due to composite endpoints was the switch study. I think almost everybody in this audience is very familiar. This was a change from chronic transfusions to hydroxyurea in secondary stroke prevention, and the study was designed to show a 12%, to accept the 12% higher recurrent strokes in the hydroxyurea arm. But they coupled with this with reduction of iron overload. And by study design, iron overload reduction was by chelation in the transfusion group, but once they were randomized to the experimental arm, which in this case was switching to hydroxyurea, they had to undergo phlebotomies. And the study was stopped not because there were statistically higher number of strokes. Yes, there were higher number of strokes in the hydroxyurea group, but it did not meet the acceptable limit, but it was stopped because the phlebotomies did not work in reducing the iron overload. So that was the reason for failure of this study. So, in summary, now, pain is a bad endpoint. Again, I would argue that this is not to say that pain should not be studied in detail. Yes, it should. It's a complex phenomenon with frequent neuropathic components. and an example of this is many payer studies. But again, maybe we should revisit the design of the studies. As important as targets downstream from sickling are, it may be better to conduct these studies in conjunction with an anti-sickling agent. And of course, again, the final point that I would make is taking a single target in an established ongoing vaso-occlusive crisis is risky simply because our understanding of the pathophysiology of this is not conducive to selecting one optimal target in this situation. So with this, what are the challenges in 2013 and beyond? Well, I think it seems like a combination therapy with hemoglobin F induction is going to be possible in the near future. We have a number of therapeutic targets on which there's a lot of study. We will hear about stem cell transplantation. There are studies that are ongoing to be able to improve that. And there's a gene therapy study that has been successful in 2 patients in Europe, and Philippe Lebouche is trying to open an arm in the US both for thalassemia and sickle cell, but I think that's several years down the road still. And the other one, of course, is improving access to care and reduce disparities, especially for adult patients. And that's the theme of our P20 grant from National Institute of Minority Health and Health Disparities, along with my colleague Robert Gibson. It's clear that Jim and I represent an endangered species. There are not too many adult hematologists coming from down who are interested in benign or non-malignant sickle cell. I'm following Pablo's comments that he made that PI3 kinase, etc., looks like cancer. Sickle cell is a pretty bad disease. By no means it's benign. So we have to form partnerships with primary care providers and provide them with guidelines or work with them closely to improve the care of this growing sickle cell population with a lot of organ dysfunction and damage. And one of the integral parts of this is improving ED care for sickle cell disease, which is dismal and which is the source of most disparity and discrimination, as we well know. And in terms of pain, studies like Kalpana-Gupta's investigating neuropathic and central mechanism, pairing with pain researchers, genomics of pain, and development of novel therapies beyond opioids to attack these mechanisms that we will decipher is going to be very important. And with that, I stop and thank you.